Development of association genetics, or is sometimes called association mapping or LD mapping, was driven by human geneticists seeking to characterize the genetic basis of disease. The approach was made possible by the discovery of literally millions of SNPs in the human genome and the development of high-throughput platforms that provide SNP genotypes simultaneously for hundreds of thousands of SNPs. The approach was rapidly adopted by plant and animal breeders who envisioned its application for marker-assisted selection, or MAS, M-A-S. The forest genetics community initiated association studies in 2000 and has contributed significantly to the development of high-throughput genotyping capacity for the plant community. Association genetics is defined in much the same way as QTL mapping is defined. That is, it is a process for identifying a statistical relationship between alternative alleles at a given locus or loci and alternative phenotypic states. We shall review, once again, the distinctions between QTL mapping and association genetics momentarily, but it is important to note that there are two rather different approaches to association testing based on how individuals are grouped for analysis. The first is often referred to as case control, in which individual test subjects are grouped by phenotype. The human genetics literature is abundantly populated with such studies. The alternative approach is to group test subjects by their genotypic classes, much like we described for QTL mapping. In part, the distinction is a function of the genetic basis of the trait of interest. Most traits in crop species are not bimodal, that is, they are quantitative in nature and do not lend themselves to being either or, that is, case control. We will discuss both approaches here, but the latter is certainly more typical in forestry and plant studies to date. There are also alternative approaches to selecting markers for testing. This was an important distinction a few years ago, but it's becoming less so as technologies improve and prices come down. That is to say, as genomes and our gene space become increasingly easy to sequence, it is becoming easier to find and test SNPs in virtually all the genes in an organism. This topic also will be discussed again further into the module. This slide illustrates a very simple association genetics experiment based on a case control example. Case control studies are straightforward conceptually, but they can lead to erroneous conclusions if not conducted properly. Designed for binary traits such as a disease, individuals are divided into two groups, diseased or healthy, and then are genotyped for an array of markers. In this case, the marker showing association is located in a gene that confers resistance to blister rust, disease in white pine species. Two alleles exist in this case, the S allele, which stands for susceptible, and the R allele, which stands for resistant. For each marker, a statistical test is conducted to see whether the two groups differ in frequency of marker alleles. Those markers with a significant test statistic are then examined more closely, with the interpretation that there is a putative causal association between marker and trait. In this case, there was an obvious connection indicated by a chi-squared test. Let's return briefly to this slide, which we introduced in the previous module. We noted that association genetics requires many markers and that ideally those markers are obtained from exons, introns, or regulatory regions of known genes. Though the size of the study population is similar for association genetics and QTL linkage mapping, the constitution of the population may be and typically is quite different. In early forestry studies, association populations have been created that represent both A, large unrelated sets of individuals, and B, dozens of small, loosely related families. Because association genetics relies on historical recombination, and strong LD that extends over relatively short distances, the associations that are found tend to be very precise in their identification of genomic regions responsible for the differences noted in the traits of interest. Of course, to find the actual genes responsible, vastly more markers are needed, and/or a prior knowledge of which genes may be responsible must be known. With a comprehensive study, genome-wide coverage with markers, that is, an association genetics test is more likely to detect the majority of genetic factors controlling a trait than is a single-family linkage-based test, especially when speaking of rare alleles with large effects. From an applied science point of view, however, by far the most important distinction between the two approaches is that statistical associations found in an association genetic study are applicable across families and may provide insight into the biochemical and genetic basis of the traits under study. This slide should look very familiar. For fear of sounding repetitive to a fault, let's just review each of these topics briefly. It should go without saying, though we do anyway, that a successful association genetic study requires each of these elements to be met with the highest standards possible. Constraints to meeting these standards are typically money and time, the latter reflected often in lack of patience and forging ahead with suboptimal populations, etc. What is meant by an appropriate population? For most forest trees, it probably means that one should pay particular attention to 1. The size, and 2. The structure of the test population. Simulation studies conducted by Long and Langley in 1999 suggested that association genetic studies use at least 500 unrelated individuals for detecting associations. Determining appropriate population size for studies using pedigrees is more complicated. Knowing in advance whether individuals chosen for a study exhibit population structure is perhaps impossible, but a number of techniques exist to analyze data and remove those effects. The importance of population structure has borne out many times, especially in human association studies. Frequent examples exist in the early association literature of associations found in one study not holding up in another study conducted with a different population. Think again about those factors that contribute to the creation and breakdown of linkage disequilibrium. The take-home lesson here is that detected associations must be verified in 2 or more populations to have predictable utility. To elaborate a bit more on how one might develop an association genetics trial in plants, consider the flowchart shown here, as modified from a paper by Flint Garcia et al. An association study will likely begin with the construction of a population and the identification of a set of SNP polymorphisms that represent the population of genes of interest. This flowchart indicates alternative approaches to identifying the collection of SNPs, namely the candidate gene approach or the whole genome approach. The order of these 2 steps, population and marker development, is not critical and may be done simultaneously to save time. For trees, identifying and establishing the population will be time-consuming and should be started as early as possible. The next step is to determine if population structure exists in your association population. This is done with an independent set of neutral markers, such as SSRs, or microsatellites. Finally, as the population matures, phenotypes of the traits of interest are obtained. You are now ready to determine if individual loci affect your traits. The equation shown here implies a mixed model analysis that includes a number of components, namely C, the effect of marker alleles, K, a measure of kinship or relationship among individuals in the population, Q, a measure of population structure, and E, an error variance. Okay, let's look at some population examples that have been used in recent forest tree trials. This loblolly pine population was originally created to study clonal genetic and silvicultural effects, but has also served as an excellent test case for SNP association discovery in populations with kinship structure. 32 parent trees were crossed twice each amongst themselves to create 64 full SIB families. From each family, 20 seed were grown up as hedges to permit clonal replication of the genotype by rooted cutting. Over 1,400 clones were created, of which approximately 1,000 were ultimately established on 6 field test sites distributed throughout the southeastern United States. This population was subsequently evaluated for drought tolerance, disease resistance, growth, and wood properties. The upper left figure denotes the distribution of parental origins by state. The lower right photo shows a small hedge plant from which cuttings were taken to root. The middle photo shows the scope of effort required just to create the hedge for all 1,000 clones. Field tests typically included at least 4 replications with 2 cuttings per clone per replication, providing a very good measure of phenotype. The advantage that perennial plants, like trees, have relative to annuals is that these field trials may be used for years to measure any number of phenotypic traits and test against the known genotypic database. We have alluded to the fact that measuring trait phenotype must be done very well to ensure success of association studies, whether they be family or population-based approaches. In the tree breeding module, we also talked about indirect selection, measuring surrogate traits that are genetically correlated to the actual trait of interest, because the latter is too difficult, expensive, or time-consuming to measure. Here we try to convey how all these features may play out in an effort to get back to selecting on the most direct measure of all, mutations in genes controlling the trait itself. Consider physical wood properties such as specific gravity, or wood density. Specific gravity affects wood strength and paper quality. Typically, the higher the density, the better. It is a valuable property. Historically, specific gravity was determined by drilling out a core of wood from older trees and using a water displacement method to measure it. The method was inexpensive, but neither precise nor very accurate. And waiting for trees to get old was, well, tiring. Very precise and accurate, but expensive methods like X-ray densitometry, shown in photo to the left, have been developed to provide specific gravity measures on a ring-by-ring basis, even distinguishing between early or spring wood and late or summer wood. This could be done for reasonably young trees without destroying them. In the photo to the right, we see a device that takes measures of the speed with which sound travels through the wood of standing trees. From this, one can also determine specific gravity and other measures of wood properties. It is fast and non-destructive, though less accurate and precise than densitometry. Still, performed on clonally replicated studies, it is yielding QTL and associations that are validated. The take-home message here are that phenotyping is as important as any other element of an association genetic studies, and that finding markers that can be used as direct measures of these traits could save a great deal of time and money in the future. Introductory papers on association genetics in plants and trees made a distinction between the candidate gene and whole genome approaches. This distinction was driven, in part, by the extent of linkage disequilibrium in the species being studied, and in part, by the availability of sequence and SNP discovery. The approach to association in human genetics followed the whole genome approach. That is, they sought to obtain a very large set of distributed SNPs that covered the genome derived from whole genome sequence. Keep in mind that linkage disequilibrium in humans is relatively large. Throughout the first decade of the 21st century, sequence for trees was relatively minimal, with few exceptions, i.e. poplar. Furthermore, conifer genomes are huge, 10 times larger than humans, for instance, and obtaining whole genome sequence for them was both prohibitively expensive and technically not yet practically feasible. How then could association genetics be evaluated in trees? An alternative approach was to seek associations of markers located in targeted candidate genes only. Early studies began by resequencing small sets of genes known to control important functions, like lignin and cellulose synthesis, drought and hardiness, and so forth. As granting agencies increased levels of support, it became feasible to conduct SNP discovery in many, if not all, known expressed genes in a species. As of the time of this writing, the end of 2010, the largest conifer studies were evaluating 5 to 20,000 SNPs, representing perhaps 7,500 to 10,000 putative genes in what we would call a non-targeted gene approach to association genetics. With the pending whole genome sequence of several conifers and flowering trees over the next few years, the distinctions made here will gradually become irrelevant. What will not change is that trees in general will still possess small linkage disequilibrium, and it will be impractical, even if technically feasible, to flood the genome with anonymous SNPs. It may be possible, however, to evaluate 2 to 4 SNPs for every known gene. How many SNPs are enough? This is a difficult question to answer and will vary for every species depending on the genome organization, level of linkage disequilibrium, and so forth. The following cartoon illustrates the relationship between the extent of linkage disequilibrium, LD, and the resolution of association studies. In Figure A, the amount of LD, shown along the y-axis, declines slowly with increasing distance along a chromosome from the gene responsible for the phenotype, identified here as a red oval. In this case, even a low density of markers, as shown as red vertical bars, is sufficient to identify associated markers, yellow arrows. In Figure B, LD declines very rapidly around the causative gene, and a much greater density of markers is required to identify an associated marker. Most trees will fall within the B category. As the table implies, it would be impractical to use the whole genome scan approach for forest trees, for far too many markers would be required. Seeking markers in only the expressed gene space would significantly reduce the number of markers required, and there are additional approaches to reducing this number further, as will be discussed in the following slide. So how many SNPs are required for a genome-wide association study? Obviously, for cost-saving purposes, the answer is the fewest possible to get a reliable result. This figure suggests ways to reduce the number of SNPs used from all possible SNPs in a human association genetics model. All common SNPs, those with minor allele frequencies of greater than or equal to 5%, are shown for one of the hap-mat ENCODE regions, ENM010 on chromosome 7, for the Caucasian population. An ENCODE region is an area identified as obtaining functional genetic elements. The term refers to the Encyclopedia of DNA Elements, a public research consortium project. All genic SNPs from this set are also shown. 4 approaches to capturing information about the SNPs in this region in genome-wide association studies are illustrated, each using a different type of genotyping SNP set. Quasi-random SNPs from the Affymetrix 500,000 array set are shown, as are 2 sets of tagging SNPs, which are chosen on the basis that they are in linkage disequilibrium with other SNPs that are not genotyped. These tagging SNPs are selected to capture all SNPs at or above pre-specified thresholds of R², which is a measure of linkage disequilibrium. Finally, the genic tagging sets consist of the SNPs that are required to cover SNPs that lie within genes at or above an R² threshold of 0.8. It is certainly conceivable that whole genome sequences will be available for a handful of important tree species within the next 5 years, but obtaining them will remain costly and technically challenging. For some of these species, though not all, association studies will benefit from the new gene and SNP discoveries. What about other species that are of both economic and ecologic importance, but for which large expenditures for sequence and SNP discovery are as yet unjustified? Such species may still benefit from association genetic studies that identify key genes for adaptive and economic traits. A logical approach is to leverage what has already been learned in the model organisms, or species, and to restrict application to only those candidate genes that have been defined to date. This would likely require some genomic work to identify homologous genes in the species of interest, characterize the species-specific SNPs for a defined subset of candidate genes, and then to verify association in a population created specifically for that purpose. The number one recommendation that can be given to tree breeders or land managers working with species other than those for which genomic resources exist is to establish appropriate association genetics populations now in anticipation of genomic resources becoming available in the future. Ultimately, identifying which candidate genes to evaluate in your population can be done in one of several ways noted here. In addition to those candidate genes that are identified in association tests of primary forest tree species, one may use other approaches, such as those illustrated here. Positional candidates are those genes with known function that map in the near vicinity of a QTL. The functional candidate gene approach requires that someone has identified a gene in another species, for instance Arabidopsis, that has a known function, and that gene can be used to fish out a similar or homologous gene from your species. Expression candidate genes are those that are found to be up or down regulated in studies where treatments are imposed, such as cold hardiness testing or drought stress testing. In practice, 2 or all 3 of these approaches may be used in unison to pick candidates. In forest trees, the candidate gene approach has almost always yielded a relatively high rate of success in identifying statistically significant associations with traits for which they were chosen. We wrap up the genotyping element of association genetics by mentioning potential issues or pitfalls associated with genotype data. The first relates to quality of data, typically obtained from outside contract labs. Virtually all base calls are done automatically in high-throughput systems today. This output should be checked visually where QA/QC suggests potential problems. Care should certainly be done when using rare SNPs. While these may very well have real effects, they are as likely to cause spurious associations just by chance alone. If retained, they should be thoroughly validated. Finally, a huge concern with association genetic studies relates to missing data. Genomic scale studies almost certainly will have a lot of missing data. This may be due to poor DNA from a given individual, poor marker design, or a bad genotyping run. Simply throwing out all records with some missing data can so weaken a study that nothing can be learned, but imputing missing data can lead to erroneous interpretations. Many methods have been developed to improve accuracy of imputed data, but the best solution is still to optimize marker DNA quality and avoid missing data altogether. Choice of statistical tests for marker trait associations is dependent on your experimental conditions. For case control studies with qualitative traits, simple chi-squared tests may suffice. For quantitative traits, regression is preferred for biallelic markers, simple ANOVA for markers with multiple alleles, and more complex mixed models where population structure that leads to confounding linkage disequilibrium needs to be controlled. Still, other methods exist for special cases, such as the non-parametric method of Kruskal-Wallis. Statistical tests of association should not be confounded with statistical procedures designed to identify and characterize population structure. The latter may produce results that can be used to make association testing more accurate. We spoke about population structure at some length in previous modules. Many factors may contribute to a structured population, which in turn can lead to detection of false positive marker-trait associations. One can see from the list above that geographic origin appears twice. This is worth some thought. What we seek are associations between alleles and measurable phenotypic traits, as expressed in common environments. The strict interpretation here is that allele by trait associations discovered using test subjects that represent two very different environments, for instance alpine versus lowland, would be misleading and a function of population structure. Indeed, it could be, but it is also possible it might be a real association of practical value. The second reference to geography, being that of isolation, refers to instances such as observed in many forest trees. Take loblolly pine, for instance. It is very likely that the current natural range of the species reflects the post-glacial migration of forests from two distinct refugia, one in Mexico that fed the forests west of the Mississippi River, and one in Florida that fed everything else east of the river. During the long separation of populations, it is quite possible that inbreeding, drift, and subsequent population admixture could yield false positive associations. Co-ancestry is a very important factor to deal with, particularly when association populations are derived from pedigreed families. However, structure arises, it is key that it be quantified and accounted for in subsequent statistical tests of associations. To illustrate how population structure can lead to misleading results, consider this example taken from Marchini et al. In this case, population sampling was highly skewed for the two conditions, case control, of a qualitative trait. The authors describe it as follows. If the study population consists of subpopulations that differ genetically, and if disease prevalence also differs across these subpopulations, then the proportions of cases and controls sampled from each subpopulation will tend to differ, as will allele or genotype frequencies between cases and controls at any locus at which the subpopulations differ. The figure shows an example of this scenario with two populations in which the cases have an excess of individuals from population 2, and population 2 has a lower frequency of the A allele than population 1. In this example, the structure mimics the signal of association in that there is a significant difference in allele and genotype frequencies between cases and controls. Because population structure is so important in the detection of real marker-trait associations, much time and effort has been invested in determining how to characterize and accommodate it. Ideally, the best approach is to avoid it altogether by being very careful in how your populations are selected. For instance, association genetic studies might be done with isolated subpopulations that are believed to have little mixing with other populations. Of course, such conditions are probably very rare for most organisms, and in any event, one would need to test them to be sure. If you cannot control co-ancestry concerns due to admixture, then one can work with populations where it is clearly known in advance, such as with known pedigrees. Whether known or unknown a priori, structure can be addressed using appropriate statistical adjustments, some of which we will note in slides to follow. We can break down approaches to detecting and accounting for structure into 3 general classes, as noted in this slide. Some software packages, such as Tassel, have been developed that simultaneously account for structured association in populations and co-ancestry or kinship while testing for association. We will begin with discussion of family-based approaches developed largely by the human genetics community. Association mapping can produce misleading results when the study population is not homogeneous, but includes individuals with different genetic backgrounds. Family-based association tests, commonly referred to as TDTs or transmission disequilibrium tests, do not produce misleading results in these circumstances, but they are designed specifically to deal with qualitative or binomial traits, such as in the case control examples noted earlier. An extension to the TDT approach called QTDT, or Quantitative Trait Disequilibrium Test, can use all the information in a pedigree to construct powerful tests of association that are robust in the presence of stratification. QTDT incorporates variance components methodology in the analysis of family data and includes exact estimation of p-values for analysis of small samples and non-normal data. The genomic control approach to detecting population structure is based on the premise that structure will affect the entire genome similarly. Patterns of variation among some subpopulations should be reflected across the entire genome. If no structure exists, patterns of variation among genes/markers drawn from throughout the genome should occur at random. To test for this, genomic markers considered to be neutral, that is, not influenced by selection, such as microsatellites or SSRs, are used to test for patterns of variation within the association population selected for study. If patterns are found, they can be used to adjust critical values for test statistics, so tests are valid. Adjustments made as a result of genomic control are population-wide and therefore rather coarse. The basic approach for population-based methods to account for structure is to decompose populations into unstructured subpopulations and determine associations within those smaller homogeneous groups. The program structure, introduced in an earlier module, is a very common way to accomplish this. Structure uses a set of random markers, such as noted in the previous slide on genomic control, to estimate population structure, defined by the parameter Q. Based upon allelic frequencies in this array of markers, group affinities are identified and the number of groups is estimated as K. The group affinities are used to allocate proportions of each individual's genetic makeup among the K groups. By allowing for individual specific adjustments, structured association provides a much finer filter for detecting association than does genomic control. The program structure does not perform association tests, but identifies homogeneous groupings within which association testing should be performed. Structured association is computationally demanding. One drawback of accounting for structure is that the true genetic basis of associations may be clouded, leading to a false negative result from association testing. It is a fine line to walk. Consequently, one may wish to not entirely discard associations that are marginally significant or those that are lost following structure testing until your results are validated. Alternative approaches to determining the presence of population structure that are computationally simpler, faster, and easier to use are traditional multivariate applications such as principal component analysis, PCA, are multidimensional scaling, MDS. Multivariate methods are based on covariance of allelic frequencies among marker genotypes. The absence of population structure would imply that the covariance structure is uniform for all individuals. Population structure would perturb this uniformity in ways that allow natural groupings of individuals to emerge from the data cloud. Mixed linear models, perhaps best illustrated by the now commonly used program TASSEL, developed by Bradbury et al., use estimates of population structure and kinship obtained by other methods to improve the ability to test for true marker-trait association. They do not rely on any particular model of population structure. In cases where known pedigrees were used, traditional mixed model analyses made kinship adjustments based on co-ancestry matrices, defined by G, determined by breeders. Today, markers can actually provide a more accurate measure of true relative kinship. So think about it. Markers can be used to both estimate kinship and population structure, and in the process contribute to the end goal of identifying the independent association of other markers with traits of interest. To give you a better sense of how the unified mixed model is constructed in TASSEL, it is shown here in linear form and with a hypothetical example of how matrix algebra would be used for association testing. Each element in the matrices represents an individual test subject. The trait mean y is described by the 8 variables noted here plus the error variance. Variable 1, chi, is a matrix of fixed location or environmental effects, and beta is a vector of site means. Variables 3 and 4 identify allelic state and effects, one locus at a time, and variables 5 and 6 identify the subpopulation from which the samples came and subpopulation mean effects. Variable 7 represents degree of relatedness, that is, a kinship matrix, and 8 is the polygenic effect generated by other loci that are unlinked to the one being tested. Factors 2, 4, 6, and 8 are all vectors. The next slide is an illustration of how the results of association trials may be visualized. This figure is called a Manhattan plot, which is a common way to visually evaluate the significance and location of markers, SNPs in this case, in a genome-wide association study. Each colored region depicts a different human chromosome, from chromosome 1 on the left to chromosome 22 on the right. Significance tests are conducted for each SNP individually across the genome, with the -log base 10 p-value for each test plotted by the SNP's physical location. Several significant peaks are shown. Notice that significant SNP associations occur in clusters. This reflects the fact that a cluster of neighboring SNPs are likely to be grouped together, perhaps in one or a few haploblocks. It's worth pointing out that by this method alone, we cannot identify which, if any, of the significant SNP associations is or could be a causative QTN. Accomplishing that task is challenging and requires further tests. One last point. A plot such as this resembles, in a general way, a highly condensed QTL plot across the entire genome, but instead of curves, only points are shown. And thinking about it a little more, we might realize that this approach is essentially identical to the single marker QTL strategy we discussed in Module 9. The major difference here is that the number of analyzed markers is perhaps hundreds of times larger. While this method may have limited utility in forest trees today, it is reasonable to assume that genome sequences for conifer and hardwood species will be available in the very near term, and with them access to tens of thousands of SNP markers. Another method used to visualize large-scale association tests is the QQ plot. Interested viewers may wish to look at the article by Pearson and Manolio, published in 2008. As a tool for genetically dissecting complex traits and providing a foundation for marker-assisted selection, association genetics offers a great deal. Though we have alluded to a number of pros and cons of the approach, we will conclude here with just a few thoughts. In many ways, these points are made relative to the efforts and rewards of alternative approaches, like QTL linkage mapping. Let's start with populations. Association genetics can be conducted on existing pedigreed materials or on unstructured, unrelated populations. In theory, this precludes the need for making specific crosses. In trees, making controlled crosses can be expensive and time-consuming, or even prohibitively difficult, depending on the species of interest. Association genetics results in very precise mapping or identification of putative loci responsible for causal associations. This has both basic and applied significance. For the tree breeder or resource manager, the most important feature of association genetics is likely to be the scope of inference derived from a test. Associations found in test populations should have a high probability of utility in a wide range of other populations or families. This approach is not without fault, of course. To be fully useful, association genetics requires a very substantial investment in genomic resources in terms of SNP discovery, validation, and genotyping, not to mention population development. Careful attention to potential confounding effects like co-ancestry and population structure are required to obtain meaningful data, and even then, one may not be able to quantify genotype by environment or epistatic effects. The latter factors can significantly influence whether associations can be verified as repeatable, making marker genotypes predictive enough to use reliably. In short, association genetics is a powerful tool, much like QTL linkage mapping, but with blemishes that need tending. In the next section of modules, we will look at pilot study attempts to apply the approaches we have discussed in forest genetic settings and offer suggestions for how marker-assisted selection may be conducted in a tree improvement or landscape environment.